Hello, and welcome to the National Association of Plant Breeders webinar series, How to Breed New Plant Varieties, Imagining and Engineering Crops. My name is Shelby Ellison, and I will be serving as your host for the webinar. Today's presentation will be given by Professor James Kelly from the Department of Plant, Soil, and Microbial Sciences at Michigan State University. Dr. Kelly leads the Dry Bean Breeding and Genetics program at MSU. His research group focuses on the development of high-yielding, disease and stress-resistant bean cultivars with upright architecture, and improving canning quality in 10 commercial seed classes for production in Michigan. Today, Dr. Kelly will be speaking about bean breeding and genetics in his presentation, Dry Bean Breeding Isn't a Dry Topic.
Well, good afternoon. My topic this afternoon will be on dry bean breeding, and the title is Breeding Isn't a Dry Topic. Let me start by introducing dry beans to our audience. Dry beans belong to the species and genus Phaseolus vulgaris, and these are the dry seed product of beans. Phaseolus vulgaris includes not only dry beans, as you can see in the slide here, the different seed types and color types and so forth. It also includes horticultural types that we know as snap beans or garden beans, shown on the bottom right of the slide. We call them dry beans because obviously they're harvested when they're mature and dry, and sometimes we need to call them dry beans just because bean sometimes gets confused with soybeans. So before I get into my topic, dry beans, as it says here, are a pretty package of health benefits. And let me just mention a little about the health benefits of beans. All beans need to be cooked, obviously, before they can be consumed. And here I'm showing a nutritional profile of 1 cup of cooked beans. And you can see that beans are high in protein. That protein blends well with cereal proteins for a vegetarian diet. They're very high in fiber, both soluble and insoluble fiber. And then we have high levels of nutrients, including magnesium, potassium, and calcium. Those are listed there on the slide, along with micronutrients, iron and zinc. And there are programs underway to enhance some of the micronutrient content. They also are very high in folic acid, which is an essential nutrient also, and they're a gluten-free product. As we look at beans, Phaseolus vulgaris, or the common bean, the common bean was featured by the U.S. Postal Service in 2006 among their stamp collection of crops of America. And here you see beans listed with their 2 sister crops, the corn and squash, which are some of the traditional crops of America, part of the rather famous milpa production system. Beans are also featured in the one on the back of the $1 Native American coin in 2009, and you can see there that their bean or corn and squash are being planted, and beans are being planted in that also. So you can see they've been very much a part of the culture of the Americas. When we look at the genus Phaseolus, there's 5 domesticated species, and common bean is the most important, the most widely grown. And one of the other species is the lima bean, Phaseolus linatus. And if you look at the stamp, just under the value of the stamp, the 39 cents, you'll see a large seed type there, and that's the lima bean also depicted in that stamp. The species ranges are as found from northern Mexico to northern Argentina. It's an annual legume in the subfamily Fabaceae, and is quite variable in growth habit, seed color, and ecological adaptation, as you'll see from later discussion in my talk. It's an important food source of protein, as I've mentioned, and as a legume, it fixes nitrogen. Even though it's a short season crop, it still fixes nitrogen similar to other grain legumes. And as I mentioned, It includes the horticultural types, the garden bean, or green beans or snap beans, as also is depicted in that illustration on the stamp. So where were common beans domesticated? Well, as we mentioned, wild bean ranges from northern Mexico, Chihuahua, all the way down to Salta in northern Argentina. And you can see that illustrated in the map on the right-hand side. The seeds that you see in the center of the screen are wild beans. You can see they're very small compared to the domesticated types on the left-hand side of the screen. And you may notice that their larger beans are found generally in South America or in the Andean region, more so than in Middle America. Prior knowledge placed the original origins of beans in Ecuador and northern Peru, but that has since been changed fairly recently. And as you can see in this next slide, common beans now have been shown to originate not as an Andean crop, but as a Middle American crop. You can see them illustrated in this green area in the state of Jalisco and Guadalajara, Mexico. From there, they moved northwards up towards Chihuahua, moved down through the Isthmus of Panama, all the way down, as I said, to northern Argentina. And we have two areas of domestication of beans: the Middle American origin, as shown in Mexico in the green area, and that area in the Andes, the southern Andes, and the violet area in southern Peru. And what's interesting is you see similar areas of domestication in Mexico for maize along with beans, part of that that cultivation, that milpa cultivation, cropping system cultivation. What also is interesting, I had mentioned lima beans. There is, again, as you look at Mexico in the upper part of the slide, you can see both lima beans and common beans were domesticated in generally the same region of Mexico. But as you look at the Andean domestication regions, you can see that beans or common beans are domesticated in southern Peru, whereas the lima bean appears to be domesticated more into Ecuador or northern Peru. So as a result of domestication, we have two quite distinct gene pools of beans. The Andean gene pool, which includes mostly our larger seeded types, and then on the right-hand side of the slide, we call the Middle American or Mesoamerican gene pool, which includes our smaller or medium seeded types. These have been further subdivided based on ecological adaptation and geographic distribution and genetic differences into three separate races in each of these gene pools. We have race Nueva Granada, which is illustrated by the North American white and red kidneys on the left-hand side of the screen. You can see the white kidneys and the two red kidneys. You have the yellow beans, which come from Peru, race Peru, and then the red mottled types known as cranberry beans, which come from Chile or race Chile. And these are quite distinctively, genetically distinct from the Middle American gene pool. And here we are illustrating it by the North American types. In race Durango, we have the Pinto and white-seeded Great Northern. The Mesoamerican, which is the small-seeded types, which are the black and navy beans. And then we have race Jalisco, which is the red and pink beans. These can all be intercrossed, but there is genetic incompatibilities between the gene pools, which prevents the free flow of certain traits between some of these, between the two gene pools. If we look at where how beans moved into North America, obviously you can see in Mexico here the arrow indicating where race Durango, the Pintos, moved right up into the center part of the U.S. and are well adapted to that region. The Mesoamerican types then moved into the Caribbean. That would be the blacks and navies. And then they moved up into the East Coast and eventually up into the Midwest, into the Great Lakes area. And then the Andean beans then moved out of the regions of Colombia and Peru into Brazil, and from Brazil they were moved then over to Europe during the 1600s, and from Europe then they were moved then as part of the slave trade into Africa, and now are widely grown in more highland areas of East Africa. Those Andean beans that also went to Europe made their way back to the US and are found then on the East Coast, and many of them are many of the old heirloom or landrace varieties that you hear people talk about on the East Coast. Here's a photo maybe of a future World Cup soccer player here. This is a picture of some bean production test trials in northern Rwanda, and you can see the climbing beans here, and these are these Andean types that are very important, a very important food source in East Africa, East and Southern Africa. And again, the seed types that you find in Rwanda are very similar, as you can see, in size and color to the North American red kidney beans or the cranberry beans from Chile and so forth. And these are very, very important food source throughout, as I said, Eastern and Southern Africa. Just a few words on dry beans in the U.S. The U.S., as it indicates here, is the 6th leading producer of dry beans. Brazil is the leading producer, followed by Mexico. And production in the U.S. is largely in the northern states, since beans are a shorter season crop, or as we go west, they're found at higher elevations where long season crops are not well adapted. So you find them in the higher elevations, higher production zones of Colorado into the intermountain areas of Idaho and central Washington. In the Midwest, Michigan and the upper Midwest, North Dakota together produce about half of the U.S. production, and most of that production is under rain-fed conditions. All the western production has to be irrigated because it's essentially in the rain shadow production area. Some 20% of the crop, the U.S. crop, is exported, and the value in 2011, which doesn't change much, is listed here at $790 million. In Michigan, you can see we're the second leading producer of beans. We're the leading producer of black beans, cranberry beans, and small reds, and the second leading producer of navy and kidney beans. And I want to point out that for those who are not familiar with the crop, it is a non-GMO crop. So in the state of Michigan, for many years, for many decades, was a large producer of white beans, the navy beans. But there was a time, a need for diversification in our industry because of opportunities of export markets, but also because of changes in national production. So our program over the last number of decades then has expanded dramatically from breeding just navy beans and kidney beans to breeding a series of different classes, many of which were not well adapted here and required efforts to change both growth habit, local adaptation, and different levels of disease resistance. And along with those changes, we had to maintain the quality aspects that beans are recognized by both the processing industry and by the consumer. So I list here the Middle American seed types that I showed earlier. On the left-hand side of the screen, the pintos, the navies, the northerns, the blacks, the reds, and the pinks. And then on the right-hand side, we have the Andean beans, the 3 kidney types, and some of the minor classes like soldier beans, cranberry beans, and yellow eye beans. And the greatest genetic diversity still lies in the Middle American gene pool, where beans originated. There's significantly less genetic variation in the Andean pool, even though you see quite a range of diversity in seed color. A little on The genetics of beans, it's a diploid, self-pollinating crop. It's an annual species. A diploid where the 2N is equal to 2X is equal to 22. And we have 11 chromosomes. The haploid number is 11. We have 11 chromosomes, and those are numbered here, PV for Phaseolus vulgaris, 1 through 11. There's very little natural outcrossing in beans, so the varieties we produce are not able to produce hybrid beans as such. We're producing pure line, near homozygous pure line varieties. In terms of the information on bean genome, it was recently sequenced in 2012. Beans have a relatively small genome size of 500, estimated at 587 megabases, and there's substantial genetic synteny with soybean. Even though soybean has 40 chromosomes, there was a duplication, but there's still a large amount of genetic synteny between beans, common bean, and soybean. Looking at the MSU Dry Bean Breeding program, I've listed here the major research objectives in our program. This would apply to many programs, not only bean breeding, but other crop improvement programs. Our focus being on yield and processing quality. Those are essential criteria for us. And then we also work with disease resistance, architectural improvement for an agronomic adaptation, and increasingly we're looking at stress tolerance and drought tolerance. And then the last one I list there is grower friendliness, and the picture on the bottom of the screen illustrates two varieties, one on the right which has retained its leaves at harvest, one which has dropped its leaves. The one on the right would require the use of a desiccant, and again, a grower-friendly variety would be more likely to be the one on the left-hand side of the photograph. Production in the state of Michigan is located in the central part of the state, up through what we call the thumb of Michigan, as you look at the back of your left hand, and that's the major production zone for dry beans in Michigan. In terms of the breeding systems that we work with, the main breeding system that we use in our program is the pedigree breeding system to focus on yield and quality traits, but we have used a series of other breeding methods as needed. Backcross breeding to introduce disease resistance, a number of years ago to improve architecture, as is illustrated in the photo on the right, to develop an upright type from a more decumbent prostrate type. We used the system of recurrent selection. We have used inbred backcross methods to introduce diversity from the distinct gene pools. And over the years, we have developed some markers for marker-assisted selection, largely for disease resistance, and I should say for qualitative disease resistance. Like most breeding programs, then, we have focused on more QTL analysis for quantitative traits in more recent years, and as we develop more marker technologies, particularly SNP markers, now there's a number of projects now starting to look at genome-wide association mapping to improve quantitative traits such as drought tolerance, insect resistance, certain disease resistances, and also improving biological nitrogen fixation. This is the bean breeding pyramid in which we kind of structure our program. We're expected to produce varieties for our growers here in Michigan or for beyond the borders of Michigan. So in order to develop varieties, new varieties will surface from the apex of that pyramid, which we largely use our pedigree, a system to develop those varieties. But to undergird that, we need a broad genetic foundation in the base of that pyramid. And that's where many of our graduate students would be working on projects in terms of genetic inheritance of specific traits, on QTL analysis, or initiating some of these studies on GWAS as a way of introducing new variability, understanding it, and integrating it into these different seed types, and then moving it up through the different systems of breeding through the intermediate level to the apex level, where we would be focused largely on yield improvement within the constraints that I list here. Constraints of growth habit, maturity. We work with a shorter season crop and with certain disease resistances, and then the quality traits that are very critical. Market class traits for the sale of the seed within the industry, seed traits in terms of size, shape, color, and then canning quality. Since all beans have to be processed, canning quality is a very is an essential criteria or trait as part of our breeding program. So looking at the breeding timeline for variety development, when we're asked how long it takes to develop a variety, we indicate generally we're working on something like a 10-year program. For the first few years are involved in our initial crossing, early generation selection. As I indicate, we do a lot of single plant selection through the pedigree system in the early generations. And to hasten the program, we conduct a shuttle breeding program with winter nurseries in Puerto Rico. Then in the second trimester, years 4 through 6, we're dealing with yield testing and replicated trials, and everything that goes into yield trials is evaluated for canning quality. And then as materials would emerge and look like promising as new varieties, then we're still looking at 3 years of seed increases from breeder through foundation and certified before we could market a new variety in a 10-year time frame. If we're looking at the crossing of beans, or here we're the bud pollination of beans, the photograph on the right-hand side shows an open flower. When the flower is opened, it's already pollinated, which therefore requires us to do bud pollination. And you can see on the photograph on the left-hand side, we're opening that flower bud rather carefully to access then the petals. And this is a legume flower. Therefore, when you can extract the banner and wing petals, you can actually press down on the wing petals, as is illustrated in the upper slide, with forceps, and that trips the flower, and it forces the style and stigma out of the filament, away from the anthers, and it doesn't retract, it stays out. So you can conduct pollination without emasculation. Then once the stigmatic surface is exposed like that, you'll simply go to the open flower that you'd be using as a pollen source. You would trip it in the same fashion, and then you can remove the style stigmatic surface, which has pollen laden on it, and transfer the pollen simply as illustrated in the bottom portion of the slide, by hooking that style from the pollen parent, which has the pollen coated on it, hooking it onto the stigmatic surface of your female parent. And with this kind of relatively non-invasive bud pollination, we can get over 90% success rate in pollination. Pollination is conducted in the greenhouse, as illustrated in this breeding timeline. We usually are conducting our crossing in the fall of year 1. Those crosses then are selfed during the spring. So each year we're going to the field with a new group of F2 materials, and that's where we initiate our pedigree selection, or our SPS, or single plant selection. We do a lot of single plant selection because we're looking at a range of plant agronomic traits, plant architectural traits, and seed traits. And then we move that material to Puerto Rico in the wintertime, where we conduct generally mass selection for more convenience on the F3 plant rows, and those then come back to Michigan the following summer as F4 plant rows, and there we continue with single plant selection at that stage. And that's usually where we end our single plant selection, at the F4 generation. We proceed then with the second season in Puerto Rico with the F5 generation, and that material then that's selected. So we're selecting in both Michigan and Puerto Rico as part of our shuttle breeding program. Those then enter in year 3 yield trials as F6 generation materials in our preliminary yield trials, and then materials get advanced. And as I indicated earlier, materials that are evaluated in yield trials then are also processed or canned, so we can evaluate for canning quality. And then towards the end of somewhere around 3 years of yield testing, we have some idea of materials that might have certain potential as new varieties, and those are identified usually as early as that stage. And here we're just illustrating that shuttle breeding. In Michigan, we're showing some of the preliminary yield trials in the upper part of the slide, a single plant selection in the F2 and the F4, and then in the bottom slide, in collaboration with the University of Puerto Rico, then we're conducting then our mass selection of plant rows in the F3 and the F5 generation. Materials then that would be selected at the end of testing in year 6, then we enter as pre-breeder materials, and since we have certain endemic diseases in the eastern U.S., including Michigan, we establish a breeder seed source in the western U.S., usually in the state of Idaho. Our university does not have a farm there, but we contract that with local companies and growers. And then we're illustrating then 3 years of seed multiplication from breeder foundation through certified, with then a distribution of commercial seed in year 10 to our growers. I want to talk a little about some of the improvements in plant architecture that we have been involved in over the years. Here I'm illustrating the 4 plant types that you find in common bean. The determinant type on the left-hand side, which we call type 1, is a typical bush type. Many of you would be familiar with this as a garden bean type that you may be growing in your garden for horticultural, for pods and so forth. The one in the middle is the other 3 types are all indeterminate with different levels of vine extension. We have the type 2, which is an upright, more similar to a soybean type, and then we have the type 3 vine type, which is more adapted to dryland western production areas. And then the type 4 I showed would be part of the systems that I illustrated in East Africa, or parts of that milpa system in South in Central America. My predecessor, Dr. Adams, developed what he called a bean Ideotype, which was published in 1982 here, and he shows a diagram of what he felt we needed for a new bean Ideotype. And you can see it's very much an architectural improved type, an upright type with improvements in plant height, taller bean, a little longer maturity to get a larger plant, greater stem strength for lodging resistance, and a deeper taproot. So these were part and parcel of that bean Ideotype. And we've been working on developing that in most of the seed types that we currently raise. And I'm illustrating that here now, these Ideotypes in the Mesoamerican navy and black bean races, in the Durango races, which were traditionally vine type 3 or prostrate types. We've now developed upright Pinto and Great Northern, and likewise upright small red and pink types. These types then provide us some level of disease resistance. These upright types give us what we would call an architectural avoidance to certain diseases like white mold. And here I'm illustrating an upright type in the upper left slide versus a decumbent type, which you can see is near touching the ground and would have much more incidence of white mold. White mold is caused by sclerotinia. And the sclerotinia on the bottom here is not a bad writing of my bad writing with a magic marker. These are written with the actual sclerotial bodies that you can collect of white mold from an infected field. The other advantage, excuse me, of the upright types is that we've been able to change our bean harvest from a three-step process where the conventional bean harvest involved pulling or knifing the beans when they were slightly immature and allowing them to dry and then coming back later to pick them up and harvest them. This involved a number of obviously more equipment, more labor and so forth, but this was the only way to effectively harvest certain decumbent plant types. So from that, with these new upright types now, we have been able to go towards something of direct harvest where we refer to maybe as clipping or straight cutting. Again, we have savings in labor, equipment, and time as a result of that. We get improved quality because we're not bringing soil up into the product, and it gives our farmers more flexibility in terms of what they can harvest, and it allows them to plant and handle more acres in a day. I mentioned canning quality. All beans have to be processed in some way before they can be consumed. So canning quality is very important. And here you can see a series of different seed types that have been blanched and put in a can. Now we're in the process of adding brine to put these in the retort for canning. You can see a range of types from white beans, black beans, through pintos and red beans. As we look at selection for canning quality, our selection is done almost exclusively on visual selection. Since we're handling hundreds of lines, we don't attempt to taste these. Most beans are sold in other products with different flavoring, so we're looking, as the consumer eats with their eye, we're using trained judges to visually select. And I've listed here the traits that we're looking at: whole seed integrity, color retention, uniformity of color, clear brine, and so forth. And as we advance materials, then we do more in-detail measurements of color with a Hunter color meter. We measure hydration and drain weight ratios. These are important characteristics for the processing industry, and texture with the Kramer shear press. So this is a series of the traits that we measure for quality. And the next slide illustrates an example of a bean on the left that shows good seed integrity versus one on the right where you see a lot of splitting and breakage. This is not because the bean was handled. These were handled the same prior to harvest. It's just that certain varieties hold together better and show much better seed integrity than the others. And one that breaks would be obviously eliminated from our program. Colors, again, critical. Color in black beans is very critical. Here we're showing some canned black beans, 3 varieties, and we're measuring color with this Hunter color meter. So the lower the number, the darker, the blacker the bean, and that's what the processing industry wants. And you can see a 1-point difference on the lightness scale can be detected by the human eye. And you can see the bean in the center, which is a new variety, has a lower score, hence a blacker bean, something that the industry wants. They don't want something that has washed out color when the consumer opens the can. Disease resistance, beans are attacked by a range of different pathogens. Those that have the light green color, like Anthracnose, common bacterial blight, and bean common mosaic virus, are all seed-borne diseases, and are particularly problematic for us, not only us in Michigan and the US, but also for subsistence farming systems overseas. So it's important to have disease resistance to those diseases that are spread through the seed. Diseases like rust, angular leaf spot, and white mold. Again, white mold is a very serious problem in Michigan. Rust and root rots, again, can be problematic in certain seasons. And these are some of the diseases that we work towards in terms of developing disease resistance or incorporating disease resistance into new varieties. Here I'm illustrating an example of Anthracnose, where the field on the right of black beans, which is a variety that's resistant to Anthracnose. You can see, if you look at the picture in the front, you can see healthy pods versus an older susceptible navy bean variety where the farmer planted infected seed. That field was completely infected, and you can see the lesions on the pods. Resistance to this disease is controlled by major genes. It's quite easy to effectively screen, as I show in the middle of the slide, in the seedling stage. But again, it's a highly variable pathogen, and we're looking at ways to improve more the durability of resistance to a disease like Anthracnose. Again, an example of common bacterial blight in the center. I'm showing an example of early infection on some materials, a healthy variety with the coded number in the center. And then, as you see at maturity, you see the healthy beans with the clear pods on the left-hand side of the screen, and the infected leaves and pods on the right-hand side of the screen in terms of susceptible to this particular disease. Again, these are diseases to which we have genetic resistance, and we're in the process of continuing to incorporate these into new varieties. In terms of tools to assist us in our phenotypic selection, we have gone to using marker-assisted selection. We've been involved in developing markers over the years for many of our major disease resistance genes here. And as I illustrate in this slide, here we have a series of Anthracnose resistance genes to which markers have been developed both in our lab and in other labs, to the CO genes for Anthracnose, the BC genes for bean common mosaic virus, or the UR genes for rust resistance. So we have markers, and the markers I'm referring to are generally scar markers at this point in time. They're very user-friendly, and they've been widely deployed by breeding programs, not only in the US and North America, but in other countries, to develop resistance to Anthracnose, common blight, and angular leaf spot, as is illustrated in this particular slide. Again, marker-aided or marker-assisted breeding has been important for these diseases, and I just list them here again, more to illustrate the names of the pathogens which I hadn't included in an earlier slide. And we have, over the years, been able to develop pyramided resistance to both rust and BCMV by using combinations of marker-assisted selection and direct selection, where we've been able to combine these to get multiple levels of resistance in both pintos and Great Northerns and navy beans for resistance, as I indicate, to rust and to bean common mosaic virus. More recently, with financial assistance from the BeanCap project, the project has developed a gene chip, which is listed here as the Bark Bean 6K chip. It's a chip of 6,000 SNP markers. This is our first access to SNP markers, and one that was very much, the BeanCap was very influential in helping us develop. Since the community was very used to using scar markers, part of that also was at North Dakota State University, they developed in a whole series of indel markers that could be used by breeders in small programs, and there's over 2,600 indel markers now also developed for use by bean breeders. So this is some of the more recent transitions that have occurred with the assistance of bean breeding. This has allowed us to expand some of those technologies in our international programs. And here I'm just illustrating an example of this is some of our work through Feed the Future Innovation Lab, known as the Legume Innovation Lab, where we're working to improve disease resistance and productivity in some of the Andean varieties. And we've been able to move some of those marker technologies to local programs and allow them to develop, again, more disease-resistant materials for their local production area. And then similarly in projects with the same funding in Rwanda, again, where beans are very much a crop that's grown by the women of the women heads of the households, and also marketing it in the marketplace. Again, developing more disease resistance in the climbing beans, which are particularly well adapted there, has been a part of our effort too in these international projects. I'm going to finish there with a reminder that beans are a nutrient-dense food. They're good to eat, or regardless of whether you call them beans or frijoles or whatever. They're good to eat, they're good to enjoy, and certainly encourage you to consume more beans. Before I finish, I'd just like to illustrate then some of the funding sources. First of all, I want to recognize the MSU Bean Breeding and Genetics team, my colleagues here, all the graduate students that worked in the project, my predecessors who have contributed to bean breeding not only at Michigan State and beyond, our funding sources through Michigan State University, and also international funding through USAID for the Legume Innovation Lab and the Bean Cap, which has helped move beans into the genomic era. And for those of you interested in some additional reading materials, I have illustrated some websites here which you could access. The Bean Improvement Cooperative is a voluntary organization run by those interested in bean improvement, breeders, geneticists, pathologists, whoever, where we provide quite a bit of information on the genetic aspects of beans and so forth. And then some of the Bean Cap videos that have been developed on plant architecture and plant breeding science, creative and problem-solving videos on the importance of plant breeding that has been developed as part of Bean Cap. And then finally, I just want to point out that the bean genome paper was released about a week ago in Nature Genetics, and it's finally indicating and providing information on those of you interested in more of the work, the sequencing of the bean genome, and some of the diversity and some of the gene pool diversity and the domestication interests and aspects of beans. So with that, I will end and turn the microphone back to Shelby.
All right, well, thank you very much, Dr. Kelly. Very enjoyable webinar. So to all of you, now we have some time to answer some of your questions.